Hello again and welcome to part 4 of the Introduction to Vaccine Epidemiology. My name is Rene Nájera. I am an epidemiologist and I am also the editor of the History of Vaccines website. Over the last 3 sections we talked first about a little history, and you can see the link in the description below. You can see the link to all the sections in the description below. Then we talked about some basic concepts, things like virology, microbiology, immunology, and even some chemistry. So go check out that section if you want to learn some basic concepts. In section number 3 we talked about community immunity, formerly known as herd immunity, or still known as herd immunity in some circles. But we talked about community immunity and how having a high level of vaccination in a given population can reduce the chances of outbreaks within that population. And today we're going to talk about how we know that vaccines work, because yes, vaccines work. It's a fact. They work. Once we go through all the steps of testing the vaccines, getting them approved, getting them out into the public, and then looking at them, they work. And so I'm going to talk to you a little bit about how we know that they do work. All right, so vaccines obviously don't come out of a vacuum. Obviously, some sort of research went into the vaccine, into developing it, into sending it out into the public, into following up on what it's doing for the public or to the public. And how do we know that they work? Well, vaccines don't start just out of nothing. They started probably with either some research in animals or some computer modeling in which we looked at the vaccine. We looked at the kind of immunity that is created after somebody has the disease in the natural way. And by natural, I mean like they actually acquired it somewhere. We looked at the kind of immune response. We looked to see if somebody can get the disease multiple times. If they get it multiple times, is it because it's a different strain, or is it because the immunity totally wanes? That is, you get exposed at some point in time, your immunity goes up and then it goes down and never comes back up. Or do you get exposed, your immunity goes up and stays up for a long period of time, such as the case with measles or chickenpox or diseases like that. And we do these models, and we look at animal models, and we see, okay, little mouse, so you got the disease and we know that you build up a good response. Now we're going to give you the disease in a artificial way, usually in some sort of exposure. And so we know exactly how much of the disease, how much of the germ, how much of the virus we need to give you in order to trigger an immune response. So we are not trying to get the animal sick. It's just giving it enough so that they have an immune response, and then seeing if that immune response will protect them at a certain level against getting the disease. Of course, we now in modern days we have the technology to look at all these things, because years and years and decades of research have been put into mathematical models that can help us understand immunity a little bit better. So once we know what happens with an animal model, usually mice, maybe some sort of ape, or some other animal, we go into what we call phase 1 clinical trials. This is the initial testing, or the initial testing in humans to see, if is a vaccine safe. So here we have between 20 and 100 volunteers who participate. We give them the vaccine, but we just want to see how it is absorbed, how it's metabolized, how it's excreted. Is it safe? Do any of these people have some sort of like allergic reaction or some sort of weird reaction to the vaccine? How does the body deal with the vaccine, with the things that go with not just the pieces of the virus or the bacteria, or if it's a whole virus or a whole bacteria, but also the preservatives and other things that go with the vaccine. This takes a couple of months to complete. We just are looking for safety. We're looking for any kind of bad reaction. And if we have a bad reaction, then that's where it stops. We go back to the drawing board. We head back to the animal models to see what happens. But if the results are good and everything's good to go, it looks safe, we go on to phase 2. And usually, on the average, about 70% of all drugs, including vaccines, get through phase 1. Phase 2 takes a few months to a couple of years, and here we're talking about a couple of hundred patients, or 300, 400 patients. So we're talking about hundreds of patients. And I don't mean to say patients, I mean to say volunteers. These are randomized trials where one group gets the drug and another one gets a control or a placebo. So this is an actual randomized clinical trial, and again, one group will get the vaccine, and the other group will get a control. And we're not going to necessarily say placebo, because a placebo is something that has no effect. For example, if we already have a safe and efficient vaccine, but we have a vaccine that we think is safer and even more efficient or efficacious, we're gonna go ahead and give that new vaccine, and then the control will be the old vaccine. And a lot of people talk about this and they're like, well, you're not really using a placebo. It's because it would be unethical to randomize people into a group that is not getting a vaccine. If we know that the old vaccine is safe and efficacious and effective, then we are not going to just say, okay, well, you're not getting the vaccine. We're just going to give you something that we know is a placebo and may not protect you. No. That would be unethical. We want to give the old vaccine versus a new vaccine. Of course, if the new vaccine is completely new, there hasn't been anything else out in the market for that particular disease, then we go ahead and we do give a placebo of some sort. Usually, these studies are blinded. And so, you don't know who's getting the control and who's getting the placebo until the very end of the study when they're unblinded and the data analysis happens. And that way we prevent, for example, somebody knowing that they got the vaccine and maybe in their head they have it that, I'm going to get a rash, I'm going to get some sort of a bad reaction. It manifests itself physically, or somebody knows that they got the placebo and so they're afraid of contracting the disease, etc. So we just want to blind these studies so we know what's going on. Again, about 1/3 of by this time, we had 70% at the beginning. By now we have 1/3 of all the studies going through of all the drugs. Going through phase 1 and phase 2. Once phase 2 is, we see that yes, the vaccine is safe, the vaccine is effective, it's giving people immunity, then we go on to phase 3, which is the big one. So in phase 3, we have thousands of people that are participating. They're volunteers. They've been recruited into these studies. These studies usually take years to complete. Here we have, again, another kind of RCT with blinding as well. And this is done over the course of a lot of years, and FDA is supervising this as well. An IRB, an Institutional Review Board, is supervising this as well. Pharmaceutical companies are reviewing this as well. Sometimes public health agencies get involved in this as well. And competing pharmaceutical companies are looking very closely at whatever vaccine you're creating because they want to know if it's successful or not. So after years of testing, after doing it in a randomized clinical trial way of comparing a vaccine to a control, if everything looks good, it's safe, it's effective, it creates immunity, it protects people against the disease more than the control would. Everybody was well informed of what they were participating in. All the adverse events are reported. FDA looks at the data and the data are clear. The IRB says yes, everything was done ethically. The pharmaceutical company submits all the information that is requested of it. If public health was involved, the epidemiologists sign off on it, then we're good to go. We're good to market the vaccine. And usually by this time, 70 to 90% of any drugs that get into this phase 3 clinical trial will go on into the market. So as soon as we're done with phase 3, we go into the market, and that is where we go into phase 4. Phase 4 is actual post-marketing analysis of what is going on. So this is where public health gets involved a lot because we are looking at the vaccine. We are looking at how many people are getting it. We are looking at the rates of the disease. Are the rates going down? If they're going down, are they going down more on the vaccinated than on the unvaccinated? We're looking at safety. We're collecting data from things like the vaccine adverse event reporting system. We're collecting data from physicians who are saying, hey, all these people who are getting the vaccine, they're not sick. Or on the contrary, if physicians are starting to report adverse events on their patients who got the vaccine, then we take a look at that as well. Pharmaceutical companies who are competing, they take a look at this as well because again, if they're competing, they want to see, if their competitor has dropped the ball or if something's going on with their product. So this post-marketing happens as soon as the vaccine enters the market. We start collecting those data. We start analyzing it. This is for the long run. This is for many years, and this is as long as that vaccine is on the market. All right, so you heard me talk about efficacy. You're saying that a vaccine is efficacious. That is the adjective that we use. And when we are talking about efficacy, we are talking about in the lab situation. So when we're doing the clinical trials, when we're doing the phase 2, phase 3 clinical trials, we look at efficacy. We're going to talk about in the real world, and that's effectiveness in the next slide. So to calculate efficacy, we look at the attack rate, and the attack rate is equal to the number of people who are sick divided by the population or the number of people at risk, right? And so let's say that we're looking at a vaccine against the flu, and the number of people who got sick was 100 in our clinical trial of 300 people, and this is among the unvaccinated, and again, this is the unvaccinated. And in the vaccinated, the number of people who got sick, hopefully we have similar numbers in vaccinated and unvaccinated in this clinical trial. But let's say that about 20 people got sick. And we divide that by the 100 over 300 again for the unvaccinated. So we can say that the efficacy of this vaccine was 0.33 minus 0.07 divided by 0.33 again. And that efficacy turns out to be 78% or 0.78 or 78.8% efficacious. Of course, there are some vaccines that are not as efficacious as others, as we will see, and there are also some vaccines that are very efficacious in the lab setting and the clinical trial setting, but when it comes to the real world, they show up to be not as effective. And again, this is because these are ideal clinical conditions where we pick the people who are going to be participating in the study. Usually they're very, very normal people, average height, average weight, no comorbidities. They're otherwise healthy. But in the population who's getting the vaccine, well, that depends. That varies. And so in those cases, something might have happened that the vaccine is not as effective in the real world. And so those are two big distinctions that we need to make. One other thing, when we're talking about safety, and that'll be in the next session, a lot of the information that is collected during the clinical trials and even before that in the phase 1 trials, that is what goes into the package insert. And that doesn't mean that those things are necessarily going to happen, and we're going to talk about that, but know that information that goes into the package insert comes from the clinical trials themselves. They come from the lab situations. They don't come from the real world situations. For that, we have other information that is available out there. On the other hand, effectiveness, this is in real life. This is in a real life situation. This is when we give the vaccine after the phase 3. It goes out to market and it goes out to the public. This is during the phase 4 analysis of the vaccine. This is where we look at effectiveness. And how do we do it? 1 minus the odds ratio. Where do we get the odds ratio for? We get it from outbreak investigations. And so when we do an outbreak investigation, we look at who's getting sick and who is not. And we look at the odds ratio and do this 1 minus the odds ratio. So remember, in the previous example, we had a 78.8% efficacy. So what is the effectiveness? So let's look at that. So let's say that we have this outbreak investigation, and over here 97.19, this is for the MMR, the effectiveness, and this is for the influenza a couple of years ago. So 1 minus the OR for it to equal 97, then the OR should have been 0.03. And so that 0.03 said that people who were vaccinated had a 3 in 100 chance of being in an outbreak, or they had 97% less probability of being in an outbreak than people who were not vaccinated. 1 minus the OR equals 19, or 0.19, then the OR must have been equal to 81, right? 0.81. And so that is a very high probability of being in an outbreak, given that you will be given the vaccine. And so we've talked about influenza vaccines. We've said that it's not as good as it should be, but it's still cutting your risk by 19% of being in an outbreak, in an epidemic. So it's still something. It's better than nothing. But here's how we calculate outbreak odds ratios. We have this cell here called A, B, C, D. Odds ratio basically is A times D divided by B times C. So let's just plug in some numbers. Let's say that people who were vaccinated, 10 of them in this outbreak at an institution like a school or whatever, 10 of them were sick, but 90 were not sick out of a total of 100 who were vaccinated. Unvaccinated, let's say that it's a very small number, so let's say that 20 were not vaccinated. But out of those 20, how many were sick? Let's say 10 as well. And notice I did that on purpose. It's the same numbers of sick and vaccinated and unvaccinated. The difference is, of course, the proportions. And so we do the odds. In this case, for the vaccinated, the odds is 10 over 90. For the unvaccinated, it's 10 over 10. So you can see this is 0.9 and this is 1.0. So the odds ratio, I'm sorry, not 0.90, it's 10 divided by 9. It's 10 divided by 90, which is 0.11. So 0.11 divided by 1 is 0.11. So our odds ratio is 0.11, meaning that if you were vaccinated, you have about 89% less odds of being in the epidemic than if you were unvaccinated. And you can see we can do our little formula: 10 times 10 divided by 90 times 10, which is equal to 100 over 900, which is equal to 1 over 9, which is equal to 0.11. And so when we're looking at the effectiveness, in this case, we can say that this vaccine was 89% effective at preventing you from being in that epidemic. And that is a very important distinction to make. It's not saying that it's going to reduce your chance of getting sick by 89%. It's saying that in an outbreak situation, you're 89% less likely to be in the outbreak. People seem to hear effectiveness or efficacy and associate it with their personal chance of getting the disease being that they got the vaccine. No, it doesn't work that way. We're talking about population level events. We're talking about population level measurements. And so we're talking about an 89% reduction of you being part of the outbreak. If you were at this school when this outbreak happened, the probability of you getting the disease out in the public or out in the general public or at home or whatever is much different. For these 90 people who were not sick, the probability was 0, right? They all got the vaccine and they all got not sick. For them, the probability of getting sick was 0. For these people here, the probability was 1 in 9, right? Or 11%. So it was slightly different. When it came to not being vaccinated, they had a 50-50 shot of being sick or not sick, which is it's much higher than the 1 in 10, right? So again, this is something you need to remember, that this is not looking at your personal risk of getting the disease. This is looking at a population level of being in an outbreak. So let's look at some examples of some outbreaks out there. This is a measles outbreak in Minnesota a couple of years ago. There were a total of 65 cases reported. The age range was between 3 months and 49 years. Remember that in the United States, you have to get your first MMR at 12 months of age. So there were some kiddos who were not able to get the vaccine, and thus they were classified as unvaccinated. But because they were so young, we don't put them when we do the calculations for effectiveness, we don't put them in the unvaccinated. We put them in the other category. And so 62 out of 65 were unvaccinated, 95%. Look at that. So if you were not vaccinated, you had a really good chance of being in this outbreak. 50% of those children were age eligible for MMR. So 50 out of the 62 unvaccinated were eligible, or they were over 12 months of age to get the vaccine. 2 of the 65 who were vaccinated had both doses. 2 of the 65 who were sick had both of the doses, and so 97% effectiveness, just like we always hear about the vaccine. It's 97% effective. So this is kind of showing us that even though you get vaccinated, there's slight, very tiny chance that you'll be in this outbreak again, because the vaccine is not 100% effective. It's not 100% efficacious. And then 55 of the cases were U.S.-born children of Somali descent. This was very interesting because a known anti-vaccine person, somebody who created a fraud around the MMR vaccine, went to Minnesota and talked to these parents, these Somali immigrant parents, and said, I'm going to continue this. Well, he didn't say this verbatim, I'm just paraphrasing. But he continued the fraud and continued to say that the MMR was associated with autism, when all the evidence is not there showing any kind of association between autism and vaccines. So this is an outbreak of measles in Minnesota. You can read up on it. It's very interesting because it kind of exploded very quickly, and it exploded because, again, measles, the R naught is what? Around 16. Right? And now let's look at this outbreak of whooping cough. Pertussis is the scientific name. Bordetella Pertussis is what causes it. And it's very bad. If you see a child with pertussis, you can tell that they're having a rough time because they just can't stop coughing. The toxin attaches itself to the breathing, the little villi, the little hair-like structures in the breathing tubes and the trachea, and you start coughing because you're producing a lot of mucus. Well, in this highly vaccinated, very, very highly vaccinated school in Korea, they still had an outbreak of whooping cough. Total of 9 cases, or about 1% of the students. What does that tell you? About 99% effective, right? They were 7 to 10 years of age, 95% of the school children fully vaccinated. Again, very highly vaccinated, but you still had an outbreak. But this outbreak, look at this. One case had full-blown pertussis. The other ones had mild symptoms. The other ones had mild symptoms because they were vaccinated. The toxin was still attaching to their villi, but their immune system was acting quickly and trying to get rid of that toxin and getting rid of the bacteria as well. So the symptoms were milder. Only one case had full-blown pertussis. Guess who was the unvaccinated one? And so you'd say, well, oh my God, you have this outbreak of whooping cough, even though 95% of the children were vaccinated. But look, in schools with lower vaccination rates, up to 75% of the children may become infected. So in other parts of Korea, in other epidemics that they've had, other outbreaks that they've had, 75% of the children were sick with whooping cough. Here, 1%. So that just tells you that, yes, again, even though you have a population that most of them get the vaccine, you're still going to have outbreaks. But those outbreaks will be made up mostly of unvaccinated, or in proportion of mostly unvaccinated children. The unvaccinated children will have more of the full-blown disease than children who are vaccinated and still part of the outbreak. They will have milder symptoms. And if you have lower vaccination rates, you lower herd immunity, you up the chances of epidemic, and you have up to 75% of the children who become infected. At the very end of the series, you will have the citations to all these studies, or you can just ask for them and I'll put them in the comment. All right, so something that happened in 2009 during the H1N1 pandemic in 2009 was that people were saying, you rushed this vaccine to market. You didn't test it. You didn't do clinical trials. What is up with that? Why did you not go through phase 1, phase 2, phase 3, and then eventually phase 4? Well, there wasn't really a need to do that. Let's just say, for example, that apple pie over here is the traditional H1N1 vaccine, non-pandemic. The one that was always being given up to 2009. To make this apple pie, we have sugar, flour, eggs, spices, and butter that go into it. The only thing that is different is apple. When we have the pandemic with the H1N1 strain, we still made our pie with sugar, flours, eggs, spices, and butter. The only thing that we changed was the cherry component. And that cherry component was just another strain of H1N1. For all intents and purposes, those two strains were the same, except for a tiny little piece of RNA code that allowed this H1N1 pandemic to get by our defenses because the antibodies were not matching up exactly with the virus. And so when that happens, then we have a pandemic situation because most of us don't have immunity. The vaccine didn't need to go through phase 1 and phase 2 and phase 3 because we already know how to make the pie. We already know what is going into the pie. The only difference was that little bit of flu from this new strain. And scientifically speaking, there was no difference. You were not going to have any kind of bad reaction to the vaccine if you would not have had one to the previous vaccine, because all that was being changed is the strain. And this is not only between the non-pandemic and pandemic, this is a year to year as well. So year to year, from one season to the other, this is what happens. We just go ahead and use the same ingredients to make our overall vaccine, and then we just add either apple or cherry depending on what depending on what virus is circulating that year. So this is why the vaccine for the pandemic, it was not rushed to the market, it was rushed to production so it could go into the market. But they still took some time. They still took a couple of months, and they did get some people together. And it was a small group. It was maybe a couple of hundred people who were given the new vaccine with the cherry pie filling, just to be absolutely sure. But they didn't do a full-blown phase 1, phase 2, and then phase 3 study because this was an emergency. Everybody in the world, by the definition of a pandemic, was getting influenza. And so another thing that public health does is that we look at the overall decrease in morbidity and mortality. And for this, we're looking at morbidity, which is the number of cases of a disease in the United States once the vaccine goes online. So before the vaccine, for diphtheria, we had about 21,000 cases of diphtheria. After the vaccine, 0. We had about 29,000 cases of smallpox. After the vaccine, 0. And in fact, we haven't had any cases in the world since 1979. Congenital rubella, which was really, really bad. Children being born with congenital rubella syndrome with deformities, 152 every year. After the vaccine, 1, if that. There's been a lot of years with 0. Polio. Remember polio? No, you don't, because we haven't had a case of polio in the U.S. in forever. So now we're at 0 for that. Varicella, which is chickenpox. 4 million cases every year. A lot of kids getting sick, staying home, parents not able to work, having to watch them at home, going to the doctor, paying medical bills. Now that's still around, but not as much and not as bad. Tetanus, 580 cases. Lockjaw, people paralyzed. Now 8, maybe, if we're quote unquote lucky. Pertussis, 200,000 cases a year of children coughing so hard that they would break their ribs or they would suffocate. Now about 20,000. Still some work to do there, guys. Measles, 500,000 a year. Now 61. That's climbing. You know, we're getting now into the hundreds because of anti-vaccine sentiment specifically against the MMR vaccine. Hepatitis B, 66,000 cases before the vaccine. Now 11,000. More and more people are getting the hepatitis B vaccine. Children are being given it. My daughter got her first dose at the moment she was born. So hopefully she will mount an immune response so should she ever become exposed to hepatitis B. Hepatitis A, 117. Now we have 11,000. So you can see the trends, right? You can see that once we give vaccines to a lot of people in the general population, the number of cases will drop. And when the number of cases drops, the number of deaths drop as well. So this is very interesting to keep in mind, because there are some anti-vaccine people who will say to you, look, the number of, on the right-hand side, the number of measles deaths was dropping before the vaccine, and it was actually really low by the time that the measles vaccine came on board in 1963. So they'll say, we didn't need a vaccine because the number of deaths was dropping. And, you know, it was eventually going to hit 0 if you look at the trend. Two things. This is first of all, the y-axis that is being used. Second, these are deaths, not cases. Third, medical technology advanced, and so we are able to keep people alive when they have measles until they recovered. But the number of cases is still there. Look at the left-hand side. This is the same time range when they claim that measles was at an all-time low with deaths in the 1950s and 60s. Well, look at the 1950s and 60s here. There were still a lot of cases, and this is a number in thousands. So 500,000 cases right there, 450,000 cases right there. The vaccine comes online in 1963 right here, and what happens? Boom. The number of cases drops. And then in 1990, second dose is recommended, and it just drops to almost 0. Now we have only a couple of hundred cases. We don't have thousands. We certainly don't have 500,000 of cases. So on the one hand, looking at deaths, yes, there were other advances, and we're never going to deny that there are other advances in medical technology that help people survive through a disease. But why should they be having the disease to begin with when the vaccine has lowered the number of cases, right? So we have antibiotics, we have iron lungs to keep people alive on polio. That doesn't mean that we can allow polio to come back because, hey, you know, it's not a big deal if you get polio. We just put you in the iron lung and you'll live in there forever. No, probably not something that we want. And so, yes, other medical technology can prevent deaths, but vaccines, in addition to that medical technology, and in places where that medical technology is not available, those vaccines can go in and they will lower both the number of cases and the number of deaths. So don't let yourself be fooled by some of these charts that are floating around out there that say, look, the number of deaths was already dropping before the vaccine. We didn't need the vaccine. Well, sure, that's true. The number of deaths would be probably minimal, but the number of cases would still be in the 500,000. And why put a child through that? You don't really want a child to go through a febrile disease that has the very true potential of giving them encephalitis, or having them lose their hearing or lose eyesight, or even kill them. No. This is where the vaccines come in, and this is why we know that they work. So with all that, thank you so much for sticking around until the end of this discussion. The links for the previous parts are in the description. Part 5 will be coming out soon enough, and we will be talking about how do we know that vaccines are safe. So now we know the background of vaccines and the history. We know the basic science that goes into them. We talked about community immunity, and now we know the evidence that vaccines work. So next, we will present the ways that we know that vaccines are safe. Again, links in the description. Don't forget to subscribe so you get the updates. Thank you so much. Have a great rest of your day.